We're going to see how to recreate figure 1 from the Calour manuscript, the amphibian transfer experiment, using Easy Calour, which is the full graphical user interface for Calour. So first we will run it, just run easycalour.py, and here we have the interface. So first we need to load the data. This data is from the Bletz et al. Paper, so we need a bound table and the corresponding mapping file. So this mapping file, Calour guesses the name, we can change it if we need. It's an amplicon experiment, we'll call it Amphibians, just for convenience. So Easy Calour loaded the experiment, we have 370 samples and around 2,500 features or bacteria. So the first thing we want to do is keep only the transplant experiment. This study was made of 2 experiments, so let's filter the samples. We're in the sample tab based on the study ID, and we will keep only the transplant experiment. And let's do it. It will give the name automatically. And we have now only 292 samples remaining out of the 370. Okay, so this is nice. Again, now let's keep only the intestine samples. We have both gut samples and skin samples. So in order to do that, we'll take the swab type and let's choose the intestine first. Let's call it intestine. So now we have only 156 samples. These are the intestine samples. Note that we didn't filter away features. So now let's filter away features and cluster them so we can have only the more interesting bacteria. So we keep only ones that have that have a total of 10 reads over all of these samples, and we cluster them, meaning that similar features will be close to each other. Bacteria showing the same behavior will be close to each other. Okay, now we can look at our data and look what it looks like. So we select the plot. We will plot it by the experimental category, which indicates what sample type it was, and let's plot it. So we get this heat map, and even by looking at this from far away, we see that we see a very large group of bacteria, the group here, which is present in pond, pond to pond, pond to stream. It's not present in the stream samples. Again, it's present in the stream to pond samples, and again, not present in the stream to stream samples. So this is an interesting behavior. We can zoom in a bit and just look at this bacteria, and it looks like a very strong behavior. So it seems like things coming from pond or transfer to the pond from the pond, or in the pond and transferring to the stream, or from the stream to the pond, have this set of bacteria that other samples don't have. So let's try and now make it statistically rigorous. So we'll go to the Analysis tab, and let's do differential abundance. So we'll do differential abundance based on the experimental category. Let's look, we don't want to insert biases into our analysis, so let's look just comparing bacteria coming from the pond or pond after transfer to the pond again. That will be our group 1. Second group will be the stream, or bacteria from the stream transferred to the stream. Let's do the false discovery rate, control it at 10%, using the rank mean test. And this is what we have. We got something like 260 bacteria different between these 2 conditions. So let's look at it again based on the experimental category. And remember, we only filtered based on the pond and pond to pond versus stream and stream to stream. So we didn't put any requirements on these bacteria, how they behave in the pond to stream and stream to pond. And what we see, there is a huge group of bacteria. A lot of these bacteria are high either if you ever touch the pond. So if you were from a pond to a stream, from a stream to a pond, it doesn't matter where you ended. As long as you were once in a pond, these bacteria colonized this salamander's gut. Now, note, however, this is not the behavior of all bacteria. If we zoom in, for example, the first bacteria is present anywhere that it touched the pond. The second bacteria is present according to the last habitat. So if you're in a pond or from a stream to a pond, you have this bacteria. However, if you were transferred from a pond to a stream, you lose this bacteria. So this specific bacteria behaves differently, again, because we put the condition only on the pond and pond to pond versus stream and stream to stream. But if we look on the large picture, we see a huge group of bacteria that are affected by exposure to a pond at any time. We don't see such reciprocal behavior for the stream. So we don't see a large group of bacteria that if you were ever exposed to a stream, you have them. So this is the first panel of the figure. Now we can try and recreate the same analysis for the skin. So again, we take the study, the transplant study, take the samples, and now let's keep only the skin samples. So we filter again, swab type. This time we'll take the skin samples. let's call it skin. So we have this experiment, again, cluster the features, so similar bacteria will be the same. And let's plot this experiment now, only the skin samples. And look. And when we look at the skin samples, we see a different behavior. So just by looking at the large picture, we see a group of bacteria around here that are present in all samples that end in a pond. Either pond transfer to pond, or stream transfer to pond, and are less present in the things that were transferred to a stream at the last point. I can zoom in and look at them a bit. And we see this group here. Again, present in things that end in a pond, either not transferred to a pond, or from a stream to a pond, but not present in pond transfer to a stream. Okay, so again, we'll do the same analysis, the differential abundance analysis, again, basing it only on the samples that are always in a pond or always in a stream. So let's choose here pond and pond to pond. Second group, we'll choose the stream and stream to stream. And again, FDR of 10%, usually it's a good number. And let's give it a name, differential abundance skin. So for convenience. And we got 125 features of bacteria. And let's plot it and look how they behave. And here again we see this behavior, even though we didn't put any requirements on the stream to pond and pond to stream. We see here that most of the bacteria depend on the end location. So pond, pond to pond, and stream to pond. Not where you began with, but where you ended before we took the measurements. Which makes sense because skin bacteria are mostly affected by where you are now. And the other way around, the skin to skin, stream to stream, and the pond to stream. So this is how we create these figures in EasyCalour.